Hi, my name is Kelsey Darnell, and I'm going to be talking to you all about carotenoids. Um, the book pages that cover carotenoids and principles of food chemistry by demand, it's pages 244 to 252. Food chemistry, principles and applications by Hui, chapter 10, page 2, and chapter 25, page 12. Food chemistry by Belitz, Grosh, and Schauberl, page 18. Food chemistry by Finema, pages 673 through 681, and Introductory Food Chemistry by Brady, pages 520 to 529. Just some background information about carotenoids. They are the most widespread pigment in nature. As you may know, they are red, orange, and yellow hues. In foods, they are dark leafy greens and then colored vegetables. Some examples are spinach and kale, and those actually are green in color because the chlorophylls mask the color of the carotenoids. And then also things like carrots, sweet potatoes, tomatoes, squash are all high in carotenoids. Carotenoids are only produced by plants and then a few microorganisms, but they can be found in some animal tissues if the food that they eat are high in carotenoids. One example will be salmon, which kind of appear orange in color, and that's due to the marine plants that the salmon eat are really high in carotenoids, so that's where they get their color from. Carotenoids are also found in chloroplasts, and they play an extremely important role in photosynthesis. They are secondary pigments in harvesting light energy behind chlorophylls. Carotenoids also have some antioxidant properties in vivo, and some serve as precursors to vitamin A, which is their most prominent role. The most prominent role of carotenoid pigments in our diets and in the diets of other animals. And for example, beta carotene possesses the most pro vitamin A activity because of its two beta ionic rings. And provitamin A carotenoids in fruits and vegetables are estimated to have between 30 to 100% of the vitamin A requirement in human populations. Carotenoids are polyene hydrocarbons biosynthesized from 8 isoprene units and have a 40 carbon skeleton. And they're divided into two classes, which are carotenes and xanthophylls, and carotenes are hydrocarbons and xanthophylls are oxygenated. Some important structural characteristics to note are whether or not there's a presence or absence of oxygen, if there's an alpha or beta ion ring, the number of conjugated double bonds, and whether or not it's cis or trans formation. So now we're going to talk about some of the physical properties of carotenoids. They are hydrophobic, so they're soluble in fats and oils and acetone and ethanol. They're moderately heat stable, but they're subject to loss of color due to oxidation. And the color is actually a result of the conjugated double bond system. So the more conjugated double bonds present, the more red the hue. Next, the chemical properties. Carotenoids are very sensitive to oxygen and light, and as I just said, undergo oxidation very easily. So when oxygen and light are not present, carotenoids are generally stable at high temperatures. And oxidation is promoted with sulfites, metal ions, and lipoxygenase due to the presence of free radicals. But carotenoids also have antioxidant activity, so they're able to quench and inactivate singlet oxygen that are formed when exposed to light and air. And this quenching ability also depends on the number of conjugated double bonds. But when carotenoids are oxidized, they are actually completely destroyed. So in vivo, carotenoids serve as photoprotectors, protecting against singlet oxygen, and in vitro at low oxygen partial pressures, carotenoids inhibit lipid oxidation, and at high oxygen partial pressures, they act as pro-oxidants, such in beta carotene. So antioxidant functions in carotenoids are actually proposed to limit cancer, cataracts, arteriosclerosis, and the aging process. And there's a lot of research done about each one of these topics if that's something that you're more interested in. So now we're going to talk about isomerization. Generally, carotenoids are present in the all-trans configuration with the exception of algae, which are in the cis configuration. And large numbers of geometrical configurations can result from isomerization because of the large number of conjugated double bonds that are present. The more double bonds, the more possible configurations. For example, beta carotene potentially has 272 different cis configurations. And this is really important because beta carotene is responsible for the provitamin A activity. And when there's all trans configurations, there's 53% provitamin A activity, whereas cis there's only 13%. And they're not sure what causes this difference. So a quick note on the effects of processing. Generally, carotenoids are relatively stable during thermal treatments, but actually one interesting note is that blanching does increase the level of carotenoids because it inactivates lipoxygenase. But the most important thing to remember is that oxygen and light are the major factors in carotenoid breakdown. So if there's no oxygen or light present, they are really stable for thermal treatment.